Hello everyone, I'm Jithin Varghese, a research scholar at the Faculty of Physics and Mathematical Sciences at the Autonomous University of Nuevo Leon. Today, I am pleased to present our research on the topic Green Synthesis of Carbon Quantum Dots from Multi-Walled Carbon Nanotubes for Photothermal Cancer Therapy and Imaging. Carbon dots are a class of nanomaterials with dimensions below 10 nanometer, and they possess significant properties for biomedical applications. Among these, the important property is that it is very less toxic than the other nanomaterials. In this study, we propose a novel method to synthesize carbon dots from multi-walled carbon nanotubes for multimodal applications including photothermal therapy and imaging. Is an emerging cancer treatment. A key advantage of our method is that it does not use any hazardous reagents and the synthesis process is straightforward. So since we are not using any hazardous materials, there is a possibility to use this for the biomedical applications easily. So the synthesis method has been um, summarized in the table 1. So we employed multi-walled carbon nanotubes, terephthal and ascorbic acid in the ratio 1 is to 1 is to 1. And terephthal is a thiocyanine dye which is soluble in water. So this thiocyanine is used for solubilize multi-walled carbon nanotubes in water. Here we can see that the sample J1, J2, J3 have similar concentration. All the components were taken 0.001 gram, and so they are differ in their ultrasonication time. J1 is for 30 minutes, J2 for 9 hours, J3 for 18 hours, and J4 is having a different concentration, or we can say that it is more concentration than the other three samples, and its ultrasonication is time is 3 hours. So now we can go to the results and observations we got during this research. Here in this figure 1, we can observe the synthesized sample under normal light. So the samples J1, J2 and J3 exhibited a dark color, and also you can see that this dark color is increasing as the ultrasonication time is increasing from J1 to J3. That is due to the more dissolution of multiple carbon nanotubes in water. And also J4 is showing a bluish color that can be observed in these figures. Initially, all these samples were showing this bluish color, and this bluish color is due to the presence of terephthal. So after sometimes after ultrasonication, this bluish color is lost in these three samples, and that is because the terephthal and ascorbic acid generate reactive oxygen species. This reactive oxygen species actually help in the fragmentation of multi-walled carbon nanotubes. Since it's creating uh, reactive oxygen species, this terephthal is altered and partially degraded. So that is the reason for the absence of bluish color in these samples. The blue color in this J4 is attributed to the more concentration of terephthal. As we can see in the table 1, there is a more concentration of terephthal is there. So that the more terephthal, the extra terephthal is maybe absorbed to the particles in that solution. So since these carbon dots have a fluorescence property, uh, we uh, we could also observe the fluorescence property of these all the synthesized samples. So it is shown in this figure 2. This fluorescence is under UV radiation of 365 nanometer, and we can observe that J1, J2, J3. The brightness is decreasing here because, and this one, as in the, we can see that under normal light there is a more dark color in J2 and J3. So that is the reason for reduced fluorescence in J2 and J3. And in J4 we can see that more bright green color is there. So next we can go to the other characterizations. First we can go to the UV visible spectrum. Here UV visible spectrum of all the four samples were analyzed, and first here we can see that this black color is for the J1 and red color is for J2 and blue color for J3 and green color for J4. And we can see that there is a peak for J1 to J3. There is a peak in the UV region. And also we can see that as the ultrasonication time is increasing, there is a blue shift is happening. There is a small blue shift is there. That is attributed to the size reduction of these particles. And also there is no peak around 600 to 700 nanometer for this J1 to J3 samples, because this peak is due to the presence of terephthalate. It's a pi-pi interaction. So as ultrasonication time increases, and or the concentration of terephthalate is very less, this vibration is lost and there is no absorption in this particular region. But for J4 we could observe that absorption in that particular region. So that is due to the terephthalate is absorbed on these particles in the solution. Next, we did FTR spectra of the samples, and we got a similar spectra for all the samples, and we analyzed the functional group or the vibrational mode in these samples, and we got the vibrational mode or the functional groups as shown in this table 2. And there is an OH stretching is there, symmetric and asymmetric CH stretching is there, CO stretching is there, and CH bending is observed. And there is also the presence of CO2, that is due to the atmospheric contamination. And also, sample J4 was analyzed for photothermal performance. And this photothermal performance was done with 980 nanometer near infrared laser at 1 watt power. Despite of minimal absorption, we could observe some increase in the temperature that is almost linear. So even if we optimizing the laser wavelength, the concentration, the power, then we will get most efficient photothermal agent. And also we have done stem images of the three samples, and in this J1 we can observe multiple carbon nanotubes as such, and in J2 there is a presence of multiple carbon nanotubes with some morphological changes, and also it's visible as small particles, possibly carbon dots. And in J3 we could observe the graphene layers. Graphene are the thin layers of graphene could be observed, and also there is a presence of carbon dots are there when we filtering out this graphene and other large particles. So in conclusion, our green synthesis method for carbon dots offers several advantages over conventional synthesis methods and nanomaterials. Since the CDs are synthesized from multiple carbon nanotubes, they exhibit mechanical stability and potential for surface functionalization. This method, combined with surface modification, holds promise for significant application in photothermal therapy and imaging. Our future works will focus toxicity analysis, surface modification, and optimization to enhance the efficiency of PTT. And also, these are the some of the references. And thank you for your attention. Thank you very much.